I would like to welcome you all for this today's webinar where we would discuss about OmicsLogic training and research programs. Today we will get you all started with the introductory coursework of your field of interest and also discuss the stepwise guidelines to proceed with the training program completion to research fellowship opportunities under mentor guidance as well. My name is Sonalika Ray, and I'm the OmicsLogic Community Manager, and I'm responsible for daily interaction with thousands of users that are a part of our bioinformatics community that is managed by Pine Biotech. So before we start with the talk, I would like to introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. OmicsLogic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. OmicsLogic training and research is running internationally in 8 different regions, and over 15,000 users around the world have completed the training so far. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. OmicsLogic programs follow a project-based learning approach using research-grade tools to analyze data from top peer-reviewed journals. OmicsLogic training has been completed from 162 countries in over 300 workshops for 6 different specialization tracks, being oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Now, to introduce my team, I'm honored to have been working with a team that collectively has an amazing experience in this area, including the other co-founders of the company, Dr. Alfred Tauber, who comes from a medical background with an expertise in oncology, immunology, and biochemistry of information. And one of his major projects is the establishment of the Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people mentioned here on the right. Julia Panav, who is an expert bioinformatician and has helped prepare many of the materials that we will use. Avi Titovsky, who is a computer scientist and leads the development of the T-Bioinfo platform that we would be using. Dr. Harpreet Kaur has an expertise in the application of statistical machine learning techniques on biomedical data and development of prediction tools and databases, and looks after the curriculum development, bioinformatics, and data science course development. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer-reviewed journals and collaborations in industry and academia. Dr. Raghavendran is an accomplished computational biologist with an experience in large-scale analysis of multi-omics data and data science methods in precision oncology and protein DNA structural bioinformatics. So that was what I wanted to discuss about our team. And now I would like to pass on the stage to Mr. Ilya Brodsky, CEO of Pine Biotech, to talk about the various training programs that have been designed for the university. Over to you, Ilya.
Thank you very much, Sonalika. And thank you everyone for joining today's webinar. So we're doing this webinar at the invitation of Dr. Iman Turia, and we're excited to kind of get this started. So let me share my screen. I want to introduce you today to a platform that we developed so that we can facilitate effective training as well as a transition into research using computational biology and bioinformatics. And we hope that some of you will be able to register today and try some of the things out. So registration itself is free. I want to go through some of the basic features. So if you could please all navigate to learn.omicslogic.com. I will just kind of show you around in the beginning, and then I will proceed to discuss about some of these specific training opportunities that we will have, as well as how does this apply to actual research using bioinformatics. So I'll give it a moment. If you go to learn.omicslogic.com, you will see right here on the top, there will be a login button. And then you have to just fill out some basic details, and then you will have a profile. And this profile will collect all of the information that you will be kind of accumulating over time as you go through some of the examples that we have prepared. But also I will describe later on how you can find here some specific types of training and certification that is now offered through the Louisiana Biomedical Research Network, which covers many different, primarily undergraduate institutes across the state of Louisiana, but also is a part of a larger network of universities across the US called the National Association of Principal Investigators. And we have been contracted to facilitate some of the training in bioinformatics for the whole network. So please let me know if you had the chance to log in. You can just say yes or put something in the chat so that I know. Again, the link is learn.omicslogic.com, and I'll just take a moment to show everyone how to use this site. Okay, so the OmicsLogic portal is really designed to address some of the gaps in computational biology and data science for biomedical data using a number of different tools that we will review today. But starting from the very basic training, so if you're not familiar with many of the concepts in computational biology and bioinformatics and big data analysis, we'll talk about that. And then proceeding on to specialized applications of bioinformatics in several different domains. And so here, if you scroll down, you will see some of those that we are focused on, which is oncology, infectious diseases, neuroscience, and agriculture. And importantly, it is facilitated by our cloud-based infrastructure that is for big data analysis, as well as for coding and data science exercises that I will explain in the context of our data science training as well. Okay, so I'll just give it just another minute. Okay, so as I mentioned, the registration itself here is free. All you need to do is create an account. Once you have created an account, you can complete your profile, and that will give you additional benefits that I will talk about in the future. And especially it's important for this process of certification. So the way that this portal works is it tracks your activity, providing you suggestions on what are the next steps for you to go to. And as a result of completing either the synchronous training that is just available on the portal, or some of the programs that we will discuss in a few minutes, you will be able to get the certificate. And depending on which university or organization you're from, it is going to be adapted to that as well. And after you have completed the basic profile, you also have the opportunity to work on an independent project. And so this is in reference to what Sonarika already mentioned, a transition from basic training to actual research. So after completing your profile and following the directions, which includes accumulating a certain number of points, 10,000 points, which you get for different kinds of activities, you're able to then submit a project proposal and design a computational biology or a bioinformatics project from identifying a data set to performing your analysis and finally submitting somewhat of a poster presentation or essentially a preparation for an article. Now, the benefit of this process is that along the way, you have training materials to explain every stage. facilitate kind of training and practice of the different methods, and also get feedback from our expert network that will be able to evaluate your project. And today, at the end of today's session, I will describe how you can use some of this information in the end when you actually want to present your research to others. Okay, so first of all, about the basic training, what kinds of things can you actually find on here? So there are several different sections here. When you go on the portal, you will be redirected to a page for courses, and you can just simply search for courses, courses that include some very basic things like bioinformatics in Python, or specialized things like a description of a project data set with some of the methods that were used in the publication that you can try to perform yourself and compare your analysis results with the analysis that the authors did. So these are organized by several different tags, including tags by data type, like transcriptomics or genomics, course or a project. So if you're interested to try a project, you maybe already know the basics of bioinformatics, you want to apply it to a specific type of a data set, you can try that. And then also we have different types of levels, either more technical, like coding in R and Python, or more user-friendly, where you can use our T-Bioinfo data platform that allows you to process data and analyze it without the need of coding. You can also find the same topics kind of highlighted below. These are essentially collections of courses dedicated to a specific type of data, or an overview of different applications that you can use. And then finally, there are also programs. And one of the things that we recommend to those that don't have a background in bioinformatics is really to highlight this Getting Started button. The Getting Started button essentially gives you an overview of 3 important topics that you can cover in a short period of time. And then you will be introduced to some of the coding practices in R and Python. So Introduction to Bioinformatics is a free course that you can complete maybe in an hour or so. Really gives you the applications of bioinformatics that you can evaluate from healthcare to agriculture. Bytes and Molecules, which is an overview of some of the basic terminology and some of the introductory materials to topics like transcriptomics or genomics or metagenomics. And then an Introduction to Bioinformatics in R and in Python are really some introductory materials to get you started with this challenge of coding. Now, as you progress further, you will be able to build on top of these basic skills and really get into more of the advanced training that I will describe briefly in a second. Today in this presentation, we will only sample some of this material. Again, I recommend going back, but just to explain, I want to highlight what are some of the key topics that you can find on this portal. So the basic kind of training, and this is basic training that has been standardized, trying to kind of address some of the typical applications of bioinformatics, different types of data. So I already mentioned Introduction to Bioinformatics, the specific types of omics like genomics, transcriptomics, metagenomics, and epigenomics, which includes analysis of whole exome and whole genome sequencing for somatic mutations and germline mutation analysis, for example, in cancer, transcriptomic data analysis, RNA-seq for bulk and single cell RNA-seq data, metagenomics, 16S ribosomal RNA, and epigenomics. And epigenomics includes both genome-wide methylation and ChIP-seq. And then the final kind of a collection of courses is a training in data science for coding, statistical analysis, and machine learning. And this is especially focused on these different types of omics data. So we call it for biomedical data. So the different basic skills that you might gain from completing some of this training now are applied in very different domains. And these include oncology, infectious diseases, neuroscience, and agriculture. And because these domains have specific considerations about the type of data that is being generated, but also what is it implying, what is the information does it contain, we separate this into specialization tracks, essentially by using these project examples from these different domains, but also providing you with mentor-guided program resources. And so a program, and today I will speak primarily about these mentor-guided programs that are coordinated with the Louisiana Biomedical Research Network. So essentially they are provided on a semester basis. So right now we're in the fall semester, and the upcoming semester starts in January, where you will find essentially 3 or 4 programs per semester that you can choose from and get certification from the Louisiana Biomedical Research Network as a professional certificate, but also, as I mentioned, as a part of the greater National Association of Principal Investigators, which is a network that covers 23 states and Puerto Rico. So what are these programs? First of all, programs are supported by the program resources that you will already find on this portal. And that means that you will have access to asynchronous materials, big data analysis platform where you can analyze large data sets quickly, coding exercises that you can complete in your browser, and an in-depth review of project data sets that have been curated specifically around the topic of that program. And so today we'll talk about several of those examples, but I just want to again remind everyone that this is applicable to both people that are maybe more comfortable already in a coding language like R or Python, or that have no experience in coding. And for that, we have the user-friendly T-Bioinfo platform. So the objective of these programs is to explain the logic of big data analysis and be able to perform and try it, to then learn the technical skills to kind of appreciate the impact of data science in biomedical data, and then also to be connected to a network of mentors that can help you address some of the technical issues and get you beyond kind of the typical online educational challenge, which is that, you know, once you face a challenge, there's nobody to talk to. And so that's where we really provide that support to get you beyond some of these technical things. So because today's webinar is specific to Tulane University, and so we have selected some of the topics that we think are maybe the most appropriate for this audience. But again, if you explore this website, you'll be able to see some of the additional things that are covered in some of our training programs. And so the first example that I would like to start with today is COVID-19 sequencing for variants of concern. So it seemed like this epidemic has really brought to the forefront some of the applications of bioinformatics for genomic sequencing and population health. And especially we see now a resurgence of interest because of this Omicron variant. And one of the important things that we see in this specific application of bioinformatics is the link between phenotype, for example, transmission level or severity of infection, the genomic variation or variance. And in this case, the word variant doesn't mean a single mutation like we're used to in the genomics for mammalian organisms, but really means a combination of variants because these are viral mutations that accumulate very fast over time, as well as things like geolocation and time, so phylogenetic. And then finally, the kind of relationship between sequence and structure. And so that is really the objective of this training. And it is sort of based on some of the publications that have been recently in the in high impact journals that speak about the variants that are commonly studied, like variants of concern, as well as the small kind of unnoticeable variants when we talk about consensus analysis. So these are lower frequency mutations that we learn to analyze. And I will talk a little bit more about this in a few slides. Now, the second example is single cell transcriptomics. So single cell genomic data analysis and transcriptomic data analysis have become an important focus to really understand in detail what happens at the individual cell level and how do different cells describe conditions based on the proportion of types of cells. So cell type and cell biomarkers are very important there, but also change over time. And so these are the two things that we really cover in this kind of training for single cell transcriptomics. And I will be excited to share that, we have a number of tools and resources for learning some of those. And I will talk about that in detail in a little bit. The other one is, again, for data science, or R and Python for bioinformatics. So we have a, kind of realized that for biologists, one of the major challenges in bioinformatics is becoming comfortable with this way of dealing with data by learning how to code and maybe starting off with a library of scripts that you can leverage to make your analysis faster. And so in this data science training that I will describe today, this is really one of the objectives, is to explain the logic of analysis when different statistical methods and machine learning methods or visualization methods are applied to a particular biological kind of an experiment, how to run them and how to understand the results, and then to have access to these snippets of codes that you effectively learn to modify so that you can apply them to a data set of your own, and really to understand how this code works so that you understand the significance of specific portions of that code, or how to glue it together into a sequence of steps that you can then perform. And then finally, the last example that we will briefly talk about in terms of a specialization training that is coming up is structural biology, or how to apply chemoinformatics to understand the relationship between structures and compounds, and how to kind of learn to investigate different aspects of structural biology and chemical information about molecules to understand how it is typically applied in the context of different challenges that are typical for biotechnology and pharmaceutical industry. Now, as you can see, a lot of these topics rely on different types of omics data. And so because of that, one of the major things that we are going to be focused on is multi-omics integration. So one of the challenges in multi-omics integration is both the sense of very different data types of data that describe different levels of omics, like genomics or transcriptomics, and then how to combine that information when we know that really this information speaks about same biological phenomena. And so to kind of talk about all of this, I want to briefly introduce the T-Bioinfo Platform, which is essentially designed to help process as well as integrate multiple types of omics for multi-omics studies. And so to look at multi-omics, let's go to server.tbio.info, and I will type that in right here. And I'll share the link in the chat here as well. Thank you, Sparsh. Okay, so this account will actually be automatically created for you after you have created an account on this Learn portal. So after you create an account, within 24 hours, you will also be given access to this platform as well. And this is to let you analyze and interpret or integrate multiple types of omics data. But again, the training for how to use this platform is also available here as courses. And also, if you just want to get started, we have a section on tutorials right here, where you will also find a number of very short kind of tutorials to get you started. Okay, so what is this platform? So this platform was designed to help you process big data in sections like transcriptomics, genomics, or metagenomics down here below, and then to be able to analyze the relationships between features of each one of these different data types and understand them either as an integration. So right here you have data association and integration, or for data mining, where we have machine learning unsupervised and supervised machine learning. And then we also have some specialization sections for cancer biology and virology. So to illustrate how this platform works, I want to give you a brief example. And I want to highlight that under many of the sections, we have this drop-down arrow, and the drop-down arrow allows you to sample one of these projects with a demo pipeline. So the demo pipeline I will use today is right here for RNA-seq for the cell line project. And I want to, the point of this presentation is to really demonstrate that it is very user-friendly and follows a very specific logic of analysis that will be very easy to understand after this illustration. So obviously we start with this button called Start. And here you have a brief overview of the major steps for bulk RNA sequencing data analysis. So we have to first pre-process the data or prepare the raw data that we have for analysis, map the data onto the reference genome or transcriptome, and then quantify the results of our analysis. So we actually have a structured table with individual genes and numbers that signify how their level of expression varies across our sample set. So once we click on OK, you can see that I have a number of next steps that have been highlighted. And now based on my understanding of these steps, you can see the color coding. So all of the pre-processing steps are in blue. All of the mapping steps are in red. So mapping on genome or mapping on transcriptome. And the quantification is in green. And so now I have different options. So I'm going to start with pre-processing or preparing the reads for the analysis. I will also perform PCR clean, which helps address the bridge PCR amplification, which is a part of the sequencing process. And then I can either map on the transcripts or map on the full genome. Now, the difference is obviously the alternative splicing, right? How do I evaluate differences between my samples or conditions, which are groups of samples, at the level of genes, which are not typically what's actually produced into proteins, but it's the transcripts, right, with alternative junctions between exons, and that we call collectively alternative splicing. So I can either perform HiSat2 for mapping on the genome and kind of developing a novel transcriptome by constructing isoforms or how individual exons are spliced together, making a new GTF file, which I will save for mapping on the transcriptome, and then finally at the end, quantification. And so the objective of quantification, as we already said, is preparing this table of gene expression or isoform expression with a number per sample or per column right here. So this is an example pipeline where we can end or go to differential gene expression. And now we can quickly see the results. And the results, as you can see, are either gene expression or isoform expression. And so very quickly, as you can see, you can now look down and kind of remember the steps that we took because they're color-coded, they're organized into a logical workflow. And now once you've got a few examples of data sets that you can work with, you can really appreciate how quickly you can get from raw data to structured data. So now you have tables of expression. Now, once you have the tables of expression, what do you do with those tables of expression? So there are typically two major approaches. One approach is to understand statistical variation between groups of samples or differential gene expression. Differential gene expression can provide us with deep insights into biology that is represented by this transcriptomic quantified data. And for that, we need to annotate individual genes into groups of genes with pathway or gene ontology downstream analysis. And so we have over here a section that essentially combines by inputting our raw data or the gene expression data, the table of expression, to differential gene expression, and then enrichment and gene set enrichment analysis, which are downstream steps to map pathways and understand how biological processes could be evaluated through the lens of this transcriptomic data. The second major approach is machine learning, right? When we have data sets where we suspect individual samples to be grouped into new groups, right? Groups that we have no additional phenotypic information about, we are essentially mining our data to find associations between samples or even features like genes, and then try to understand what might they mean. How are they related to each other and why? And so clustering, and we have here a number of different clustering methods implemented, as well as network analysis, are typical approaches for data mining, with some visual interpretation using things like PCA. And then the second approach is when we have some kind of phenotypic information, or let's say clinical information, and we know that we have several different types of samples, maybe 2 or 3 or 4, and we would like to classify this data or perform supervised analysis. And for that, we also have a number of methods for classification and regression. So these are some of the standard methods. And of course, as I mentioned, one of the major objectives of specifically this platform is to provide multi-omics integration capabilities. And for multi-omics integration, a major challenge is not just the ability to integrate, but also to understand what are the many different approaches that exist and how do they compare. And so to briefly speak about this challenge of multi-omics integration, I wanted to invite Dr. Raghav, who will speak about his research project on patient-derived xenograft models as an example of multi-omics integration for selecting drug treatment for individual patients.
Thank you very much, Alaya. Good morning to everybody. I would now, I will share my screen and let's start. Okay, I am Raghavendran. I am a research consultant and mentor with Biotech and OmicsLogic. I'm trained in bioinformatics and I hold a PhD in computational biology from Max Planck Institute, Germany. My research interest and focus is on data science techniques such as machine learning and deep learning applied to integration of multi-omics data like gene mutation data, gene expression data, and copy number variation data. My role in OmicsLogic is developing curricula and mentoring students in the research fellowship program. Please let me talk about one such example where I developed machine learning models to predict tumor response for a variety of treatment options by employing multi-omics integration of gene expression data with gene mutation and also with copy number variation data. So we know that cancer is the leading cause of disease deaths worldwide. So for such a dreadful disease, systematic cancer treatments have relied on chemotherapeutic drugs that are marginally more toxic to cancer cells than normal cells. This is a huge problem because on an average, only 25% of the cancer patients respond to cancer drugs. But in comparison, about 80% of the patients respond positively to popular painkillers available. A prominent solution to this problem is precision oncological therapy, where a treatment strategy is designed by targeting the molecular characteristics of the patient's tumor. Hence, the treatment is personalized to patients who are likely to respond positively to the specific cancer therapies. There is not sufficiently enough data from cancer patients to molecularly characterize the tumors under multitude of different treatment options that are available. Hence, we have to resort to suitable model systems. And in this case, the model systems is patient-derived xenografts, which are immunocompromised mouse models. So we have covered why we have to do this study already. So how it is done is like this. The tumor cells from the cancer patients are subjected to next-gen sequencing techniques, and the information about gene expression, gene mutation, and copy number variation were extracted. This data molecularly characterizes the tumors, and the tumor samples that were extracted were then grafted into immunocompromised mouse and administered several cancer drugs. Then the response of these tumors to the respective drugs are documented, and these responses are then categorized into the tumors responding positively or negatively to the treatments provided. So this data records the tumor's reaction to different tumors. Both the molecular characterization data and the treatment response data are available along with the publications. So such a wealth of data generally generates a lot of research questions. My research interest in this using this data was to design and implement machine learning models to predict tumor responses to different treatments that were tried. So most biomarkers obtained to date for cancer include only one omic measurement at a time, and thus they do not take full advantage of the entire complexity of the systems captured in multi-omics data. There are, however, multiple approaches exist for performing such multi-omics integration, as you can see from here. Selecting the appropriate strategy to discover complementary information that are present in the omics data and converting them into a set of meaningful features is still a very difficult challenge. And for this particular data, I had to employ this late set of multi-omics integration because mainly other methods did not yield a very positive results and also did not provide a possibility to retain interpretability of the machine learning model. So what actually happened and what did we find? The accuracy of the machine learning model that I developed was measured as Matthews correlation coefficient, where a +1 of this correlation coefficient would mean higher prediction accuracy for the machine learning model, and -1 would mean a very low prediction accuracy, and a random prediction accuracy is considered anywhere around 0. So traditionally, traditionally, single gene biomarkers that was identified to be related to the onset of cancer was also employed to predict treatment response. This is not a very accurate model, and it has been shown in multiple different variety of ways. And I also show it in this particular way, where I apply this to breast cancer. When I use single gene biomarkers to predict the treatment response was very, very poor. But immediately when I employ multiple gene information, instead of the single gene biomarkers, the prediction accuracy of the machine learning model improves very well. And when I include other omics data, such as copy number variation and somatic mutation and gene expression data, the prediction accuracy improves very dramatically, and we get a nice set of distribution around the prediction accuracy that was observed for a multitude of different treatment profiles. So what more could be done with such a data or with such a machine learning model? One can actually extrapolate such an analysis to include patient data from TCGA repository. That would reveal us the performance of these machine learning models that are trained on preclinical data such as mouse models, but tested on the real world patient data. And if one performs that, when one performs that, one can identify the caveats, and when one can address those issues where this model is found deficient. And of course, one can perform GSEA analysis on a set of most informative genes that were extracted from the machine learning models to identify specific biomarkers that can explain the tumor's sensitivity or the tumor's resistivity to different treatments that were administered. So to perform such an analysis, coding was an integral part of my region. But if one finds coding challenging, then that should not deter them from asking such interesting questions. And this is where I think OmicsLogic Platform bridges that gap by providing not only the necessary training and also providing an alternative platform to perform such an impactful research without the necessity to learn coding in such a great detail. I thank you all for your patience, and I hand over the stage back to Elia.
Thank you very much, Dr. Raghav. And as we saw in this example, we essentially see how multi-omics integration methods are critical for classification and, in this case, prediction of treatment response. And of course, many such interesting challenges and projects could be developed. And so for anyone who's interested, on the portal, we have a number of different types of examples. For example, cancer macrophages, modeling cancer precision medicine, where similar questions about biomarkers, subtypes of cancer, and response to treatment are being asked using cell line data. PDX tumor microenvironment or patient-derived xenograft model microenvironment role in development of cancer, but also applications to other non-cancer projects like response to malaria infection, response to drought resistance in potato cultivars, and also an introduction to TCGA, the Cancer Genome Atlas. So now let's switch gears. And I want to briefly speak about other topics that I already described in the beginning, but in a little bit greater detail, because we do have an ongoing program with LSU Baton Rouge, facilitated through the Louisiana Biomedical Research Network on transcriptomics. And so transcriptomics, as you can see, all of these projects are based primarily on transcriptomic data. So there's really a lot of questions that you can ask of transcriptomic data, whether it's bulk RNA-seq or single-cell RNA-seq data. And single-cell RNA-seq data is becoming more and more interesting in the context of immunology, neuroscience, and many other different applications. And so recently we have developed a process for kind of taking that single-cell RNA-seq data and preparing tables of expression, which in itself is a major challenge to do it accurately. And so there are a number of different tools that are available on the platform just to process that single-cell RNA-seq data. But also, as anyone familiar with downstream analysis, it is also full of many technical challenges that have to do with normalization, integration of multi-sample data sets, and also downstream interpretation, for example, markers of specific cell types. And so what we have done is we have developed a course around these challenges, and now are using this to both help researchers work with their data, and we're working with a number of different groups that are using 10x Genomics as their primary sequencing platform to identify meaningful insights from single-cell data, but also for anyone who would like to learn about these different challenges in single-cell RNA-seq data analysis, we provide a tutorial that talks about processing that data and then using packages like Seurat and Monaco to understand both differential gene expression between different cell types and populations, and also discover trajectories across pseudo-time to evaluate development and specialization of different cell types. And we also are in the process of developing a number of project examples where we have, for example, this project detecting activated cell populations using single-cell RNA-seq, which is a view of different types of neurons in the mouse brain and how it could be an integration process between single-cell RNA-seq data and profiling of cell types, together with kind of an understanding of response to signal and networks in the brain. So really fascinating project. And so as we both have described, we have these different training modules that are designed to prepare you to perform your own analysis. We have different methods like processing, analysis, and integration of multi-omics data. And now I briefly want to introduce several specialization programs that are starting in January. So it's a good time to think about how this might be relevant to either research that you are planning or how it might be relevant to kind of leveraging some of these computational biology and bioinformatics skills to be able to perform this analysis and maybe even work with others to develop research projects. But before we go on, I wanted to just pause here for a second and ask if there are any questions that any of you might have, either about the project that Dr. Raghav just presented, or about what we have covered so far. And Iman, maybe you could let me know if there are any questions so we can briefly address those now.
Hi, can you hear me?
Yes.
Yeah, I'm Iman Touria, geneticist and biostatistician in Bioinformatics Department of Surgery at Tulane University. I'm glad that we all joined today to discuss this great learning opportunity for computational biology. And actually, we are dealing with a growing amount of biomedical data and with variable types, and we are in much need to analyze such data in an efficient way to reach to a data-driven hypothesis and make use of what we have formed as an output from our lab work. What we presented today, a lot of various types of platforms, in addition to integrating all of these together. This will not only provide a platform that run our analysis quickly, but also provide opportunity to learn step by step different modules and guide us from A to Z to achieve our research objectives, from designing before going into the lab and guiding us during hands-on analysis, exploring unnoticeable discoveries, till achieving valuable things as poster presentation, international publication. I started some of the platforms with the OmicsLogic a few months ago, and it was amazing. I got to know two more bioinformatic domains that was a black spot for me. It's genomics, chapsex, single-cell transcriptomics, epigenomics, etc., along with expanding my knowledge in the R and Python coding. It inspires me of more tools that I can use, and expanded my thoughts on creating more study designs and helping me in applications for grant proposals. So I really appreciate the team effort to deliver the platform in a very simple way, step by step. And I appreciate all who joined us today in this webinar. I really appreciate that the effort that is, if you really go into the website, you will look at the courses. It's clearly explained in with the screenshots, with captures, and guided by a wonderful team that at any time you can send the questions. So I really advise to go through this fellowship, or at least some of the courses. It will guide you in a lot of modules across the steps of multiple domains across omics, and will enable you to build your career at the end. Thank you, team, for making this happen to multiple researchers and students.
Thank you very much, Iman, and really looking forward to get to know some of the others here in the webinar and discussing how this might be applicable to what you might be interested in, your area of research. And so in the next few slides, and I will make it quick, and we'll just go over a couple more examples I already mentioned in the beginning, but we'll just provide a little bit more detail and kind of speak about what could be the application of that particular topic in the context of what's happening around us and what's happening in the next few months in the context of our training programs. So one of the major areas that we are involved in a variety of collaborations, and this includes collaborations with university like University of California, San Francisco, Haifa University, where the Tower Bioinformatics Research Center is, Louisiana State University, and others, is the domain of infectious diseases. And so for the infectious disease domain, applications like genomic sequencing of pathogens, as well as host response on the human or animal model side, are all very different types of applications of bioinformatics that are critical in a variety of recently kind of highlighted challenges with public health. So obviously we have the top kind of things on the list, like SARS-CoV-2, which I'll talk about in detail after this, but also some of the other kind of major challenges with emerging diseases like enterovirus EBD68, a disease that affects children, or malaria infection in response to vaccines. There have been recently novel vaccine candidates that are being advanced into later stages of clinical trials, or the recent highlights of the Ebola virus epidemic in Africa, right? How can we learn from these outbreaks of infectious diseases to really understand the role of bioinformatics And because maybe of these outbreaks that have been happening recently, there is now such a rich source of publicly available data that we can leverage to both learn about, but also perform analysis to understand and study some of these emerging pathogens. So to speak about this, I will use the SARS-CoV-2 example. And I'll mention SARS-CoV-2 as an example because also we have ongoing collaborations with a number of universities on sequencing wastewater to understand and detect outbreaks and also detect variants of concern. So for SARS-CoV-2, a number of different pipelines have been developed over the past couple of years, where primarily we focus on consensus level analysis of variants and phylogenetic, where we understand the relationship between strains. They're now categorized into alpha, beta, gamma, etc. And how are they related to time course? So how do they change over time, as well as how can we tie them to specific locations? So I think a very interesting type of an analysis, widely used in the media to describe the pandemic and some of the emerging variants of concern, including Omicron, but also importantly, it only provides us with a high level view of some of the major challenges for this virus interacting with the new host, right? So breaking out potentially through zoonotic transmission to humans and now developing into an adapted strain that is very effective at transmission, causing a very specific disease. So now as we describe this pathogen, right, one of the challenges is to also understand what are some of the ongoing processes within the host, within smaller groups of patients around the world, and how do these variants of concern emerge? And so here we have some additional topics that we speak about in this training, like detection of novel variants. So how do you determine what is a novel variant as opposed to some random mutation? How do we look at subconsensus and low frequency variants, which essentially eventually become the dominant genotype? And also, how do we deal with some of these low quality samples to understand whether we can, in a very confident way, say that there is some variant or not, and then ultimately to understand how can we map these variants onto protein structure using software like Chimera X, where we can look at the relationship between variants and their position on the spike protein, for example, and understand interaction with other proteins. And also, how can we look at the host response and understand which variants are associated with what type of a disease condition? So first of all, an easy way to get started with all of these topics is by taking a look. We have a course on the portal that I just showcased, the OmicsLogic portal, where you can get an introduction to many of these topics. So how to use some of the open data sources where you can find genomes and analyze those genomes, how to perform phylogenetic analysis to determine the origins and maybe similarity between different strains of viruses, how to understand the differences between different families of coronaviruses, how to map those variants onto the protein structure, and also how to understand the response to treatment for COVID-19 patients. So how do we understand those pathways of JAK-STAT pathway for inflammation and things like that? So it's a great resource to get started, and also learn some of these techniques using the T-Bioinfo platform, right? So before going into the coding, you deal with raw sequences, many of them. How do you integrate all of that and get easy to understand results? And so there we have developed specialized pipelines that are essentially going to produce a number of tables that you can use to visualize and kind of understand the general patterns. And then the second part of the training is really focused on producing clinical reports in R, where you will find a mapping statistics, the prominent areas that should be analyzed, for example, spike protein, and then determining and calling variants For example, you can see here variant summary for the UK 201, 20I 501YV1, right? So how do you actually perform that kind of analysis? And also learning how to code everything from the beginning to the end using the output pipelines, the output from the pipelines to produce such a report. And essentially, this is going to be useful for anyone who's involved in this kind of public health domain, or just wants to understand how is this pandemic developing over time, finding associations between where the original kind of source of infection is, how is it related to transmission, and maybe even understanding some of the response to treatment like vaccines and maybe some of the antivirals that are coming on the market. So a lot of interesting questions that could be asked. So the additional part of this is really understanding the relationship between sequence and structure. How can we not only know that this mutation happened in a specific position on the genome, but also understand the amino acids, the protein is folded. So how could we map variability found in this genomic data, and then try to associate that variability with what we know about the interaction between different proteins. So here, for example, you can see an example of some of those key mutations clustered around the receptor binding domain, as well as here we can now, we can now see how it binds to the ACE2 human receptor. And so that gives a very clear picture about the impact of some of these mutations on the typical function, right? So in this case, adaptation from one organism to another, and adaptation specifically for cell entry. So the other second kind of training, and for example, for the data science training, we have a program that we have ongoing right now, primarily for LSU graduate students and faculty, but we will have a new program that's focused on data science methods for all of Louisiana and beyond as well through the NEPI network that will be starting in January. So in January 2022, we will have an announcement made through all of the different channels that we use for communication with the specific dates. And I believe today, Sonalika will be able to share some of those dates as well. So what is included in this data science training? So in data science training, we start with just some basics in both R and Python. So how to load different sequence data, different structured data like tables, how to develop a sense of syntax, what is in the syntax. And you will find on these portal lessons that I've already shown an inline practice area. So this is a console. In the console, you get an assignment, a task. Once you understand the syntax, write it out inside this console and check for the results. And for example, here the result is multiple sequence alignment and identification of specific variants. And then in the assignment, you will have a challenge for interpretation. How do you actually interpret one mutation versus the others in terms of its impact on the sequence overall? Now, many of the methods that we use in bioinformatics and data science are packaged together effectively in these easy to call methods. And so the syntax here is not just learning how to write basic code, but also familiarity with some of the standard packages that are used to produce publication ready figures. And so in this case, just an example of several of those that could be used to develop methods that you learn how to use effectively, packages that you learn how to use effectively, and then produce the kinds of visualization that you might expect to find in a publication, and use that to both understand how the different settings are designed and how to modify them for the specific type of data that you're working with, and then make it presentable so that you can share it either in a poster, in a publication, or just an internal report. So that's really the focus of this type of a program. The program is designed around several different topics. For example, data wrangling, how to just manipulate different complex data structures and organize them, developing a sense of a preparation technique. Machine learning and statistics, unsupervised and supervised types of analysis, machine learning, how to try and compare different types of machine learning techniques and really understand why we would use one or the other in what application, and then also an introduction to deep learning. So those types of topics are all going to again be starting in January. And just to kind of briefly describe the process, you will be introduced to examples of code. The code will be, clearly explained with the different aspects of what that code syntax really looks like. And then you will be given an interactive notebook where you can perform a full assignment from beginning to the end, kind of following partial instructions and, hopefully being able to reproduce it for your own data. So, and this is just an example. So inside those lessons, you will be given those links and you'll be able to kind of go through the full code, and that will be around a specific data set that is described. A lot of different things are going to be explained there: visualization of data, different techniques for dimensionality reduction, including NMDS, PCA, and things like T-SNE, working with different types of machine learning techniques and how to evaluate their performance, including confusion matrices and ROC curves, how to understand the methodology behind a lot of these methods. What do they actually do behind the scenes? So here's an example of logistic regression, for example, right? How do we actually understand what it does, and how do we develop a sense of each one of these methods, and why we would use one or the others, and where would we find the specific settings for the way we want to use them, right? So here's an example from, how this would be implemented in a particular language, Python, with a particular library, scikit-learn. Also looking at different types of conclusions. How do you draw conclusions from these methods? Because obviously they are not straightforward, and a big challenge today is explainable AI. So how do we really explain how the model worked? Why did it work the way that it did? And what do we know about the output? How do we interpret the results? Now, I think the important part of that is practicing some of these things in the context of specific data sets, right? So data sets that have been reviewed by peers. So there are scientific journal publications, they have been reviewed, the findings have been validated, right? How do we use those examples to really understand the terminology that is used in a lot of these methods, and how do we actually start using them to find similar kinds of insights about the data? So we have these curated data sets. This is an example, cancer studies using patient-derived cell lines. In that particular paper, what the authors did was they took a collection of breast cancer cell lines and were able to integrate multi-omics data to develop a prediction score for different types of treatments measured on patients, or sorry, measured on cell lines. So how to get access to all of this training? So as Sonalika already mentioned, a lot of these programs have been designed around specific groups of students. So we've done these programs at LSU with the Louisiana Biomedical Research Network, at Georgetown University Medical Center, and even in international programs like the Amity University program in India. And as a result, we have a lot of users that have been able to kind of explore and validate some of these coursework and projects and really try them for themselves. And what we do along the way is we collect feedback from some of the users that we have, and we want to kind of share a little bit of that feedback today, just about some of the coursework. So if you have, I think an extra 30 minutes a day and you wanted to get better at your bioinformatics skills or try to use some of these skills to perform a research project, we definitely recommend that you go and just try the asynchronous version of some of the coursework that we described today. If you would really like to, quickly, with the help of a mentor, get introduced to the topic and try to understand how this topic could be used in a context of a specific research project, we recommend the upcoming training programs that we will have starting in January. So we will have a program on genomics, transcriptomics, metagenomics, and data science. A webinar dedicated to data science is going to take place this month, next week. So we will be sending out some email notifications about that. If you would like to join and kind of learn about those topics in depth, you're welcome to join that as well. We also have a special program that is called the Research Fellowship. The Research Fellowship program is designed to not train on any particular data type or method, but really to help you analyze a data set of your own or a data set from the public domain to develop an independent research project. So that is a rolling kind of a, there is not a specific time to become a part of that program. It's a program that you can join at any time and connect to some of the mentors that we have that could actually be guiding you through that process. Okay, how can you subscribe to the newsletter? Well, where all of the events and webinars are announced. The best way is if you already subscribed to this particular webinar, you probably received an email from marketing@pine.bio or marketing@omicslogic.com. And there is, thank you, Sparsh, for putting this link. We have these individual pages for different upcoming programs that we have. For example, this one is going to start on December 14th, and we have a webinar at 9 a.m. So you will be able to find this. And Sparsh, if you could find the program page that lists out all of the programs, you can share that as well. So the best thing I think is to add marketing@PineBio and marketing@omicslogic.com to your contact list, and you will see a lot of information coming from them to announce different initiatives and different types of programs that are starting. Okay, so I would like to finish because we are running over time by now, and just briefly mention some of the opportunities for research. So if you're a student or if you are a kind of starting your research as a faculty or a scientist, you can benefit from either services that our team can provide, just helping analyze your data, or joining a cohort of students and faculty that are learning to apply bioinformatics to their research projects. And so we have a lot of examples specifically in Louisiana. Yes, if we can post a link here to the Research Fellowship. So this Research Fellowship program, and I'll just go through just a couple of examples here. We've either collaborated with others on doing research for them, or you can, independently do the research on your own. I'll just share a link here for that program. It's fellowship.omicslogic.com. It's a research program, so really you already know what you are interested in, and you would like to work with mentors to understand how to perform it well. And there's a couple of examples down here at the bottom. You can also go to the main portal and see some of the example projects. If you click right here on projects, you will find other students have submitted some of their projects and what they look like. How can you contact us regarding research? So again, if this is the type of research you're looking for, it's a program that supports research projects through training. Or if you would like to have us, if you would like to discuss any research projects that you're working on and they have a challenge in data analysis, we also have a page dedicated to that as well. One second. Okay, and here you can find information about the T-Bioinfo Platform, as well as some of the services that our team provides and what that typically includes. So I hope that this is sufficient information to kind of get started. And I'm sure that we will try to organize this again in the near future to expand on some of these topics that we discussed today. So, Sonalika, are you still around? Okay, maybe not. So I think that was the main objective of the webinar today, is to just give you a high-level overview of what we do and how we do it. If you have any questions, again, I mentioned those email addresses. You're welcome to reach out to them. Or if you would like to speak to me directly, my email is ilya@pine.bio. I'd be happy to hear about your feedback or about any interest that you might have in the programs that we provide. And thank you, Dr. Touria, for inviting us. And again, as I mentioned, we're looking forward to interacting with many of you. And if you would like, yes, we do provide certificates. These are certificates that are supported by the Louisiana Biomedical Research Network, as well as the National Association of Idea Principal Investigators. And we will follow up with an email that will kind of highlight some of these additional topics that you might be interested in. So thank you for joining today. And we hope to stay in touch and do this again one time. Thank you, unless anybody has any more questions. But what I'll do is I will stop the recording.